Oh, hello there. My name is Dr. Rhys Morris. I'm one of the anatomy demonstrators here at Birmingham Medical School, and today we'll be going on a journey learning about muscle histology. Muscles are tissues capable of contraction, and they come in many different shapes and sizes. They can be broadly categorized as striated or non-striated. Striated muscle, for example, is cardiac muscle and skeletal muscle, and we'll be exploring these in a lot more detail in a little while. Before we begin this adventure though, let me introduce my sidekick and very distinguished demonstrator, Dr. Viswa Rajalingam.
Perfect timing, Rhys. Let's have a look at what I have here. Skeletal muscle is designed as a bundle within a bundle arrangement. We will start with the whole muscle and then work our way down to the microscopic level. In this image, we can see the entire muscle is surrounded by connective tissue called the epimysium. Within this, there are multiple fascicles or bundles of individual muscle cells. Bundles are surrounded by perimysium. Individual muscle cells or myocytes are surrounded by a connective tissue sheath known as the endomysium. You should also note that myocytes are also known as muscle fibers.
Before we go further, let's remind ourselves of some important features in muscle cells. The cytoplasm, or as it's also known in muscle, the sarcoplasm, contains multiple myofibrils. These are contractile elements made up of repeating units called sarcomeres. The muscle fibers have a specialized endoplasmic reticulum called sarcoplasmic reticulum. This stores calcium within the muscle, which is used during muscle contraction. The cell membrane of muscle, or as it's also known sarcolemma, has specialized invaginations into the cell known as transverse tubules. These convey the depolarization to the sarcoplasmic reticulum within the cell. This arrangement of transverse tubule and sarcoplasmic reticulum is known as the triad.
The sarcomere is the basic contractile unit of the muscle. It is composed of thick and thin filaments. Let's look at the thick filaments first. They are made up of myosin. Myosin is arranged as a dimer. A myosin filament consists of lots of these dimers with heads projecting from either end. Each myosin filament is surrounded by 6 actin filaments. For simplicity, only 2 can be seen here. The actin filaments are anchored at the Z disc, forming the ends of the sarcomere. The arrangement of the actin and myosin fibers gives the muscle its characteristic striated appearance. The region containing actin and myosin is known as the dark band or the A band. The region containing only actin is known as the light band or I band.
During muscle contraction, the release of calcium from the sarcoplasmic reticulum allows cross-bridge formation to occur between the actin and myosin, and they slide over one another. This is known as the slide-in filament mechanism and can be seen in the following animation.
Let's move on to the other type of striated muscle. This one is involuntary and it just keeps going.
The histology of cardiac muscle is slightly different to skeletal muscle, and we will now take a look at some of the differences. Firstly, cardiac muscle cells have only one nucleus. The cells are also branched, and these fibers are connected at specialized intercalated discs. These discs contain gap junctions which are crucial for function, allowing movement of ions between adjacent cells.
Another important feature of cardiac myocytes is the presence of dyads instead of triads. These consist of T-tubules and a single sarcoplasmic reticulum. The more astute amongst you may have noticed that this time the T-tubule runs along the Z disc as opposed to the AI junction in skeletal muscle. It is also worth noting that these cells have much less capacity to regenerate than skeletal muscle.
Wow, that was amazing. What type of muscles left to look at? That's right. This time it has a license to contract. Involuntarily, of course. Smooth muscle is found in many places, including lying in the gut, blood vessels and urinary tract. Smooth muscle has a central nucleus, although the sarcoplasmic reticulum is only very rudimentary and no transverse tubules are found. The arrangement of actin and myosin is very different to striated muscle. This time the layout of filaments is not organized and they have a crisscross lattice arrangement. They are inserted into dense bodies, also known as focal densities, in the cell membrane, and contraction changes the shape of the cell.
To summarize, skeletal muscle is striated with multiple peripheral nuclei. It is arranged in long fibers and there are satellite cells which allow for some regeneration. Cardiac myocytes have a single central nucleus. They are branched and are connected at intercalated discs. This time, however, they have very little regenerative capacity. Smooth muscle, as the name suggests, has a non-striated appearance. This mononucleated cell, unlike cardiac muscle, has very good regenerative capacity.
I hope you've all learned a lot from this video, and remember, boys and girls, to work hard and keep studying.
And maybe someday you too can be a part of the anatomy team.